Hello, everyone. Thank you very much for coming to my presentation. My name's Alex Charlton. I'm a toxicologist at Syngenta, and I'm going to be talking to you a little bit about the method developments for the topic of carcinogenicity from our agrochemical development perspective. So I'm going to start by talking very briefly about Syngenta as an organization. We're a global agrochemical company headquartered in Switzerland, but with employees and business interests around the world. In addition to our role as a company that manufactures and sells agrochemicals, we also have a very strong interest in research and development activities. So we've got a fairly large R&D staff and a fairly substantial R&D budget. And the way that we approach our research and development process builds safety in from the outset. So we're both interested in tools that are useful in a regulatory context, and I'll talk about that as we go, but we're also interested in tools that can supplement our research and development efforts. So before I get into the more detailed slide, I'm just going to very quickly cover how humans are exposed to pesticides. So there are three basic groups who get exposed. There are people who are occupationally exposed because they're doing things like preparing the pesticides to be used through mixing and loading activities. There are groups who are then responsible for actually spraying the pesticides. And there are also groups who have activities like re-entering into fields that have been sprayed. So there's an occupationally exposed group. There's also a bystander group, so people who happen to be traveling near fields that are being sprayed, for example, people who happen to live near farmland also. And the last group are people who are exposed via the dietary route, basically through consuming food products that have been treated with pesticides at some point in their development. We've got a fairly good level of control of exposure to people who are occupationally exposed. We can do things like set PPE requirements, telling people they must wear goggles or they must wear long shirt sleeves or gloves or whatever when they're working with our products. But when we make our safety decisions, we have to do that with the understanding that there are also going to be groups who may be exposed and whose exposure is difficult to directly control. So, for example, consumers. We do our cancer testing for two basic reasons. Firstly, there is a safety responsibility for Syngenta, where we have a corporate responsibility to ensure that humans are not harmed by the use of our products. That includes cancer. And there's also a regulatory compliance part of this. So a large number of regulatory agencies exist throughout the world whose job is to regulate the use of pesticides. And some of these agencies have specific testing requirements around carcinogenicity. Some of those are quite black and white, carved in stone. And some of them are more open to having scientific discussions about replacing studies where technically justified. So an example might be the US EPA. I'm going to talk a little bit about our current testing approaches, because I think that will hopefully help highlight where we would look for some help. There are four basic topics I'm going to cover, but I'm going to cover the genotoxicity topic very, very quickly now, and the others in a little bit more detail later. So the first way we start to test for carcinogenicity is we eliminate genotoxic carcinogens from our research and development process. And the way we do that is essentially by running standardized OECD test guideline compliant in vitro and in vivo genotoxicity studies, or cut down versions thereof. So we tend to use that a lot at the early stage of our R&D process, but we'll also use it throughout the process as we do things like change synthetic routes to ensure that we've not introduced genotoxic impurities and ahead of major studies. I'll talk a little bit about that in a second. The three topics I'm going to talk about in more detail in this presentation are our current R&D design tools, so that's in vitro screens, omics technologies primarily. I'm also going to talk about how and when we run the definitive cancer bioassays in rodents. And then lastly, I'm going to bring it all together by talking about when we do cancer mode of action assessment and how we use that understanding to refine our endpoints, our risk assessment endpoints. So this is a very stylized cartoon of the Syngenta research and development process from a human safety perspective. The process takes about 10 years to bring a compound to market from the very early stage concept to actually having a registration. And as we go, we're essentially moving from testing a very, very large number of compounds in relatively light detail to really characterizing in a huge level of detail what a very small number, typically just one compound, actually does from a cancer perspective. So as I've said in my previous slide, we do genotoxicity assays throughout our R&D process, mostly front-loading so that we just eliminate genotoxic compounds to start with, but we'll also test as we go every time we make a major change. Whilst we're testing in our early stage screening, we predominantly rely on in vitro assays. As we start to move through the R&D process, we start the switch to higher content assays. That's things like omics tools. Then we start switching towards the regulatory, and by regulatory, I kind of mean standardized test guideline compliant GLP studies that finishes up in some cancer bioassays. And then where necessary, we conduct mode of action work to understand human relevance and risk assessment endpoints. So starting talking about R&D tools. So at the outset, we're predominantly interested in two different types of in vitro assays. We're firstly interested in screening assays that give us some information about cancer hazard and ideally cancer risk. So assays that can identify potential hazards are great and they have a function, but what we're most interested in is getting an early assessment of risk assessment endpoints. So we want to try and figure out not just is a compound likely or not likely to be carcinogenic, but also what level of exposure are we expecting to be associated with carcinogenicity? Because that then allows us to start developing our risk assessment and our safety positions from a very early stage. When you look at the in vitro assays we typically use, we primarily focus our efforts towards on-target toxic effects. So if you're targeting a specific enzyme system in a fungus, for example, a plant disease, and there's an analogous enzyme system in a human, you might start doing your in vitro testing focusing on that specific system. Where we have a greater degree of trouble, and one of the areas that we think is potentially right for assay development, is prediction of off-target effects. So things that are not necessarily directly related to the activity of the compounds, its biological activity, but what else does this compound do in the body and how else might it be causing carcinogenicity? And whilst we do have some in vitro tools that allow us to not just assess a hazard, but also identify risks, at the moment, we're not in a place where we're able to quantitatively predict cancer outcomes on in vitro assays alone. And this is something that would be really helpful to us. The second type of in vitro assay we use are assays that input into mechanistic modeling. Typically, what happens is our computational modelers try to build a model, realize that they need to understand a specific, say, enzyme or reaction rate question, and then we do targeted quantitative in vitro assays to try and build that model and develop our understanding. And as we start to move forward through the R&D process, we shifted away a little bit from in vitro assays to toxicogenomic assays in vivo. The way we do this is a fairly standardized short-term in vivo rodent study, and we kind of load these studies with as many high-content endpoints as we can to try and understand what may be going on in longer duration studies. So that's things like toxicokinetics, transcriptomics, and metabolomics. And using all this information together, we're able to come up with, for a certain limited number of tumor outcomes, we're able to make quantitative predictions about what is and is not likely to be a carcinogenic dose in vivo. So when it comes to conduct of cancer bioassays, these are the regulatory studies that we must do. Typically, we don't jump into those studies. Usually what we do is build our understanding through conducting longer and longer durations, short-term assays. So we might start with a 14-day study, then a 28-day study, then a 90-day study, and then feel that we have enough understanding to set dose levels and conduct our cancer bioassay. But what's helpful as we go is that we can start to see pre-neoplastic effects in some organs in those shorter duration studies. And that's really helpful to contextualize what's happening in the longer duration cancer bioassays. So these cancer bioassays, they're conducted in compliance with validated and internationally standardized OECD test guidelines. For those who are unfamiliar, I've listed the 3 relevant ones here. They're done to GLP. Essentially, what happens is that rodents receive the test item for a substantial portion of their lifetime, and they're then examined for the formation of tumors. So for a substantial portion of their lifetime, a rat, that's a 2-year study, and a mouse, that's about an 18-month study. Typically, we have a control group and we have a few treatment groups. And what we're trying to do is develop our risk assessment understanding. So identify the thresholds where cancer and non-cancer responses sit. And as part of this, the way we design some of these studies is highly dependent upon the regulatory agency that we plan to submit the study to. Some of them are very prescriptive and require us to use very high doses. And this often results in tumor findings which are not particularly relevant to what humans are actually exposed to. If we get to the end of our cancer bioassay and we start to see a tumor response, what we'll often do is investigative mode of action assessment and try to figure out why those tumors have happened and the mode of action by which they have occurred. I'll talk a little bit about that later, but one thing I'll point out here is that where cancer responses are weak, mode of action assessment is really difficult. The stronger the tumor response you have, the easier it is to figure out why it's happened. And I think the last point on this slide is when we use these cancer bioassay data in a regulatory context, exactly what that assessment looks like changes depending upon regulatory regions. So, for example, the United States Environmental Protection Agency might run a risk assessment with those data, but the European Union might be interested in hazard identification of, is it a carcinogen? If it is a carcinogen, which classification does it warrant? So risk versus hazard assessment is quite a key component of the way that we interpret those studies after they're finished. So as far as types of tumor that most interest us, this is a retrospective analysis of about 300 agrochemicals. So you can see 309 agrochemicals in the rat, about a third get tumors. And when you look at the breakdown of the organs that those tumors sit in, 63% are either in the liver, the thyroid, or the testis. So these are really the organ systems that we are most interested in. In the mouse, a very slightly lower number get tumor, but basically very similar. And when you look at where those tumors sit, 83% are either in the liver or the lung, with the liver accounting for about 77% of those tumors. So we're very interested in liver tumors. We're quite interested in thyroid, testis, and lung tumors also. So when we do see a tumor response, the next step we undertake is to understand cancer mode of action. For 2 reasons, really. Firstly, to try and set risk assessment thresholds, so identify what the AOP is, what the quantitative linkage is between key events, and to identify where those thresholds sit, because we can use that understanding of thresholds to refine our risk assessments. And the other piece of this is to start looking at human relevance assessment. So if you see a rat liver tumor that occurs through interaction with the CAR-PXR xenosensor system, typically those tumors are not relevant for humans. And that's an example of some of the frequently occurring tumor types that we see in those assays. Because we've now started introducing in vitro and omics tools earlier in the development process, we often now start cancer bioassays with fairly robust predictions for some types of tumors based upon known AOPs, which means, though historically speaking, you began a cancer bioassay and then you found out which kind of tumors you had at the end. That's no longer the case. We typically have fairly good predictions for certain types of tumors, particularly tumors that are based upon on-target pharmacology. And when it comes to assessment of tumors, we tend to follow the IPCS mode of action framework, which outlines a really prescriptive set of steps to identify what the mode of action is and how you go about assessing human relevance. So one of the key parts that we have to deal with, and sometimes causes some trouble for us, is not just identifying why the tumours have happened, but also excluding other potential modes of action. So bringing it back to the CAR-PXR liver tumour, you need to prove that the compound is, for example, not hepatotoxic, and you're not just seeing a kind of regenerative response in the liver that's driving a tumour response. You need to exclude that as a possibility. You also may need to exclude other xenosensor systems. And that can be quite a complex undertaking, sometimes as complex as figuring out what's happening is figuring out what's not happening. So when it comes to using AOPs, this is just a very quick schematic framework of the 3 tumor types that we talked about earlier: liver, thyroid, and testis. For these, we've got fairly well-developed understandings of what's happening. So you can see kind of progression from molecular initiating events to biochemical changes to the cellular changes to the adverse outcome. I guess the key point to us here is that, as we said earlier, about 63% of rat tumors are either liver, thyroid, or testis, but that means about 37% are not in this set. So what we'd like to be able to do is develop our understanding of what's happening for those tumor types and be able to more easily map out AOPs. And then this is my last slide in this presentation. We were talking, as we've been going along, I think there's 2 basic sets of ideas that we'd had in Syngenta that might be interesting for assay development. Not to say that there aren't others. The first is that our ability to evaluate a broad spectrum of kind of non-genotoxic mechanisms is somewhat limited. At the moment, you're essentially using different in vitro assays for different modes of action, and it ends up a very complex undertaking. What we'd like to be able to use is a much smaller set of in vitro assays that allow us to assess a broad spectrum simultaneously. And this is useful to us for a number of reasons. It firstly feeds into our R&D process. It means that we can make predictions about cancer outcomes earlier in the R&D process, and if necessary, discontinue development of particular compounds if we think we have unacceptable tumor outcomes. It also means that when we try to exclude alternative modes of action, that an assay that helps us figuring out what else might be happening, and if we see only a single signal, help us to then substantiate the view that the alternative modes of action are eliminated. That might be really helpful to us. And the last thing is something we've not really talked about as we've gone through, but that's one thing that's really helpful to us in the future, may be supporting read across. So at the moment, read across is very structure based. You look at the structure of your target compound and the structure of your read across candidate, and you identify structural features that are similar. But what might be more useful to us is if we can start to identify biological similarities between target and read across candidates with an assay like this that can evaluate a broad spectrum of biological responses. That could be really helpful to us and could eliminate the need for some cancer testing. And then the last point that we come across here is something that I've pointed out earlier, which is that liver tumours are very commonly observed for agrochemicals, often because we have to go to quite high dose levels, which is what triggers the cancer response in the rodent liver. They're very rarely relevant to humans. And when we start to demonstrate that, one of the things we have to do are hepatocyte proliferation assays in the target laboratory species and in humans, and essentially show that human hepatocytes don't proliferate in the same way that laboratory species do. The issue we have there is that those proliferation assays are very hard to conduct, don't always respond appropriately to reference compounds, and therefore an improved approach to try and understand hepatocyte proliferation will be very helpful to us. So thank you very much for listening to my presentation. Thank you again to the organisers for inviting me, and I'm looking forward to discussing this in more detail with you in the future. Thank you.